Good morning, dear students. Today we are going to discuss the external and internal morphology of Selaginella. So, already in the last video we discussed the general character of Selaginella, but here one time we are recalling it. So, here division Lycophyta, class Ligulopsida, order Selaginales, family Selaginaceae, genus Selaginella. So, distribution and habitat. It is commonly called as a club moss and a spike moss. It has worldwide distribution, abundant in tropics and grows in ground and shady places. Most common species is Selaginella casuarina. Mostly the species prefer moist and shady places to grow, but few species are also found growing in xerophytic conditions, that is on a dry sandy soil or rocks. Example, Selaginella lepidophylla and as Selaginella rupstris, etc. A very few species are epiphytes, nothing but which grow on the other plant. That's a Selaginella origina. It is found growing on a tree trunks. So the vegetative morphology, external character, the plant body is sporophytic and differentiated into root, stem, leaves, licules and a rhizophores. Root. The root is young, sporophytic, is of primary root, while others are adventitious. The adventitious roots are at the tip of rhizophores. Aerial roots have developed caps and cutinized epidermal cells and a center soil. Next, stem. The stem is dorsiventral and prostrate with short erect branches. The branches are arranged dichotomously. They are also pseudomonopodia, that's a false growth from one point. The shoot apex consists of single apical cell in most cases. Right, next, rhizophore. In some species, leafless and colorless branches arise from the prostrate stem near point of branching. These grow downwards and have group of adventitious roots. They are called as rhizophores. Some scientists consider them branches and some consider them as roots, and still other consider it as an organ for protection or other function. But recently they are known as adventitious roots that have dichotomous branches at a tip. Leaves. Microphyllus are present. That's a leaves are small and a single veined. There are two types, isophilous and anisophilous. The anisophilous leaves are in pairs. They may be small. These are on inserted on the dorsal side of stem. Larger. These leaves are inserted on the ventral side of the stem. Usually the leaves near the apical portion of the branch bear sporangia. That may be microsporangia or a megasporangia and are called as a sporophylus. That may be microsporophylus or a megasporophylus. The sporophylus are usually aggregated into a condensed structure which is known as a strobili. Next, ligules. Ligule, there is a small outgrowth on adaxial side of the leaf near base. It is vestigial organ and provide water. The apex, the structure of the ligule can be differentiated into two parts, glossopodium and the body of the ligule. You can see in the diagram here. Glossopodium, it is the basal hemispherical part made up of large thin-walled cells. It is surrounded by a glossopodial sheath, body of the ligule. Above the glossopodium is the body of ligule. It is made up of many large and small cells. The function of the ligule is not well known. It may be a water secreting or a water absorbing or protective organ. According to Erner, the ligule is perhaps as a vestigial. Anatomy of the stem, leaf and root. The stem. The first layer is epidermis. Thick epidermis, thin-walled rectangular cells covered with cuticle. Cortex may many layered. Outer 2 to 4 are thick-walled cells called as a hypodermis. Below is the thin-walled parenchyma having chloroplast. Have a small intracellular spaces. Central portion is separated from cortex by a cavity having air spaces. Next comes epidermis. The cortex and the central tissue is connected by radially elongated cells called trabeculae. Very important point. They contain Casparian strips. Trabeculae are modified endodermal cells. Modified endodermal cells, we call it as a trabeculae. Next comes the stellar region. The central portion of the stem is occupied by a well-developed stele. The stele is of protostelic type. That's the xylem is present in the center and surrounded by phloem on all sides. Phloem in turn surrounded by a single layered pericycle. Pith is absent here. The number of stele is variable in different species of Selaginella. Right? It is a monostelic in some example, distelic or a polystelic. The organization of the stele is also variable in different species. It may be protostele to siphonostele. Next, rhizophore. Outermost layer is epidermis. It is of thick-walled and a single-layered cells. Beneath the epidermis, there is a cortex, that's a hypodermis, which is of a sclerenchymatous cells. Thin-walled parenchymatous region. Innermost layer is endodermis. Thin-walled pericycle is present around the vascular tissues. Stele is protostele, that's a xylem is center and a phloem is surrounded by the xylem, and here rhizophore is simple. Next, root. Outermost layer is epidermis, that is a single layer covered by a thin cuticle. Root hairs are present and arise from a epidermis. Beneath the epidermis, wide zone of cortex is present. Outer hypodermis have a sclerenchymatous cells. Endodermis is followed by a pericycle. That's a single layer, and protostele is next to pericycle. Nothing but a xylem is surrounded by the phloem. Next comes leaf morphology. It is bounded by an upper and lower epidermis. Cells of the epidermal layer contain chloroplast. On the lower epidermis are present many stomata. Mesophyll is not differentiated into pellicide and a spongy parenchyma. In the center is present a single vascular bundle surrounded by a bundle sheath. which encloses a layer of phloem and many centrally located xylem tracheids. So this is a morphology of the stem, rhizophore, root and the leaf. In the next class, we are going to discuss reproduction of the Selaginella. Thank you.